And Ali, do you have any question?
Sorry, Professor. How we can define in the slide the mesothelium and regular epithelium, simple epithelium,
Mm-hmm.
because and the staining of the mesothelium always is AgNO3
Mm-hmm.
or not? It can be different.
It can be difference. The answer for the second question, it can be difference. And based on the location of simple squamous epithelia, you can decide it's a normal simple squamous epithelia or it's a mesothelium. You should see the location of this type of epithelium.
Uh, for example, if we see the slide and we don't know where is from where that make this a slide, we
Mhm,
cannot say it's mesothelium or regular epithelium.
mhm. Mesothelium is always on an outer surface. If you see an unknown slide, you will
Uh-huh.
recognize the layers of the wall of each organ. And if you see the simple squamous epithelium on the superficial layer, you have to be sure this is a mesothelium. Or if you see a simple squamous epithelium inside of the tissue, you have to be sure it's a normal simple squamous epithelia.
Oh, thank you.
Okay, so let's start with the columnar epithelia. Share my screen. This slide was the last slide of last practice. Here you can see the simple columnar epithelia with microvilli. Do you remember this slide where we make this small summary about this slide? So let's see this slide. We will see again step by step. Look around with the smallest magnification. You see that slide has several layers with several tissues. Firstly, we have to find the epithelium, the surface epithelium on the slide. You see one surface is smooth here, one surface is smooth here, and on the other surface you see protrusions. We are in the intestine, in small intestine. Do you know how do you call these finger-like protrusion of the intestine? This whole structure. How do you call it?
Microvilli.
Not microvilli, just villi.
Villi.
Yes, villi. Here you see these finger-like protrusions of small intestine. It's called villi. And please don't mix it with the microvilli, because microvilli is the surface modification of one single columnar cell. So we will describe this type of epithelia. So we know it's an epithelial basic tissue because the cells are very close to each other, and it's a surface epithelium because the cells are on an outer surface. The cells cover the inner surface of our body. And the cells are arranged in one row, and the cell nuclei are in the same level. So it's a simple columnar epithelia. So if you have a simple columnar epithelia, we always have to check the surface modifications. And in case of this epithelia, we can find microvilli on the surface. How do they look like, microvilli on the surface? How can we recognize this surface modification? we mentioned that previously, if you see this part of the slide. How do you know? It's microvilli, not a kinocilia, not a stereocilia. Microvilli looks like under the light microscope, like a shiny, homogeneous line with a light pale eosinophilic color. That's why we know it's microvilli on the surface of columnar cells. Okay, and in the intestine has several types of surface enlarging structures. Microvilli is the modification of the cell, and villi look like also macroscopy. You can see on the surface. What else do you see in this preparation? Use this picture again. Here you see some extra cells. For example, here you see a goblet cell. This is a goblet cell here. It's called goblet. Here is another goblet cell. For example, another goblet cell here. Okay. Goblet, based on its shape, it has triangular shaped nucleus. What you see here, this is a triangular shaped nucleus of the goblet cell. And the cytoplasm looks a bubble. Why is it pale? Because it secretes carbohydrates. This will be the other type of epithelium. Already it belongs to the glandular epithelium. Glandular cells secrete a mucus, which chemically it's a carbohydrate. That's why it has pale color. How do you stain the carbohydrates? How could you stain the carbohydrates? Sorry. If we want to show the carbohydrates, we should use which type of staining method? What do you think?
Is it the pale staining?
The PES staining. Javad is here, okay. If we want to show the carbohydrates in our slides, we have to use PES staining. In case of PES staining, the carbohydrates containing cells will be PES positive, will be purple. Okay, these are goblet cells. And these roundish cells, for example, here, can you see them? These small roundish cells between of the columnar cells. Here. These round shaped cells will be lymphocytes. They are white blood cells, belongs into the immune system. Epithelium is the first defense line of every tissue. That's why we can find lymphocytes between of the epithelial cells. So lymphocytes can appear any kind of epithelium. And the last step is always describing the other types of tissues in our preparation. If you see this picture. So here we have to check the other types of tissue. A moment. So on the surface we can find this type of epithelium, simple columnar epithelium with microvilli. This epithelium is on the surface here, you see. And under the epithelium, you can find the connective tissue. Here you see a loose connective tissue. Now please write down, and later we will learn about the connective tissue. We will see the buildup of connective tissue and the main components of connective tissue. And here you see a smooth muscle tissue. This part is a smooth muscle. Here we will learn about the smooth muscle. And on the superficial part of this slide, we have a mesothelium, a simple squamous epithelium. Here this will be mesothelium. So let's see these tissues. Here it's an epithelium, and under this part you can see the loose connective tissue with vessels. Here you see a huge vessels, an artery and vein, but we will see the vessels on the 13th week of semester, here another artery. And under this part of slide you can see a more darker eosinophilic tissue, which is a smooth muscle tissue. Here you see this is a smooth muscle, and these smooth muscle cells are in two directions. Here you can see the smooth muscle cells in cross section because the cell nuclei have roundish shape, and in this smooth muscle layer the cells are in longitudinal section because the cell nuclei have a rod shape. And on the surface of the smooth muscle you can find a very thin layer, very thin epithelia. This is a mesothelium, which is a simple squamous epithelium. Because the small intestine, this part of small intestine has a peritoneum coverage, that's why it's a mesothelium. Okay. Do you have any question about this slide? So let's move on the next one. Now we have to repeat the simple and pseudostratified columnar epithelia. It's a small repetition. What is the definition of simple epithelia? Simple columnar epithelia. Please tell me the definition of simple columnar epithelia. How does it look like, simple columnar? Anybody? How does it look like, simple columnar? Oh. No. Around my list. Sultan, do you know how does it look like, simple columnar epithelia? For example.
No, sorry.
Okay, don't worry. Ali, maybe? What is this line, this green line, where the columnar cells are sitting? How do you call this line? Between the epithelium, between the connective tissue, always. No. So this is a basement membrane, yeah? It's a basement membrane. So, and the definition of simple columnar epithelium, we have one layer of cells and only one type of nucleus, and these nuclei are on one row. are in one row like this. All of these cells will be columnar cells. All of these cells are in contact with basement membrane. All of the cells reach the surface. If we compare with the pseudostratified columnar, epithelia, how does it look like? This is a basement membrane where the epithelial cells are sitting. All of the cells will be columnar cells, but the size of these columnar cells will be different. That's why the cell nuclei are arranged in several levels, several layers. And you see that all of the cells are in contact with basement membrane, but not all of them can reach the surface. Okay. And in case of the pseudostratified columnar epithelia, we have an extra cell on the basal part of the epithelium. Do you remember the name of this roundish cell? Helia? Do you remember these cells?
Isn't it reserve cell?
Thank you, reserve cells. These are reserve cells. And each one can have surface modification. These surface modifications can be microvilli, kinocilia, or stereocilia. We discussed the characteristic of microvilli. Let's see the function of microvilli. What was the function of microvilli? The function of microvilli, it's not a hard, difficult question. Microvilli looks like this.
This is absorbing the material, depends on where they are located.
Firstly, microvilli can increase the surface. That's why it can increase the absorption, for example, in the small intestine. Thank you. It's a microvilli. Microvilli is a finger-like protrusion of the cytoplasm. And the kinocilia, it's a bigger structure. The size is between the microvilli and stereocilia. It can be 5 to 10 micrometer long. And what is the main function of kinocilia? Movement. They are capable to synchronize movements. So, and the last one, columnar cells, the last one will be the stereocilia. Stereocilia is a huge microvilli, and it also can increase the surface. And how long is the stereocilia? It can be 10 to 20 micrometers long, and it also have actin filaments in the axis as microvilli. Okay, and now we have to see the options of simple columnar epithelia and pseudostratified columnar epithelia if we see the surface modifications. We can have simple columnar epithelia without any surface modification. And we have simple columnar epithelia with microvilli. We see this slide previously. And we can have a simple columnar epithelia with kinocilia. Okay. And there is no other option in case of simple columnar. Let's see the pseudostratified columnar epithelia. We have a pseudostratified columnar with without any surface modification, and there is a pseudostratified columnar epithelia with stereocilia. Oh, sorry, stereocilia. And we have a pseudostratified columnar epithelia with kinocilia. There is no other option. Let's see the example. Where can you find simple columnar epithelia without any surface modifications? In case of collecting ducts of glands, we have some ducts with simple columnar epithelia without surface modifications. For example, in salivary glands, we will see this type of epithelia. In case of, for example, in case of the collecting ducts of, it's enough if I'm writing ducts of glands, it's easier. Of the glands. And where can we find simple columnar epithelia with microvilli? So if you remember the previous slide, where do we have simple columnar epithelia with microvilli?
The small intestine.
Thank you, in the small intestine, very good. And in case of large intestine, we also have this type of epithelia. And in the gallbladder, we will find this type of epithelia. And where do we have simple columnar with kinocilia? We will see this type of epithelia in case of the female genitalia. For example, in the uterine tube or fallopian tube. You say also that, you can say also that, fallopian tube or uterine tube, or also in the uterus, we have this type of epithelia. So let's see the other type of surface epithelia, pseudostratified columnar. Without surface modification, also the ducts of glands are lined by this type of epithelia. For example, in case of salivary glands, the biggest ducts, they are lined by this type of epithelia. Okay, and let's see the pseudostratified columnar with kinocilia. Which system has this type of epithelia? Pseudostratified columnar with kinocilia. In the respiratory system, we have this type of epithelia. Okay, and the last option, pseudostratified columnar with stereocilia. There is this type of epithelia in male genital organ, for example, in the epididymis. Epididymis. Okay. Or in the vas deferens, we also have this type of epithelia. And today we will check these surface epithelium. We checked this one, simple columnar with microvilli. Okay, so and the next slide is from the uterine tube. And this slide shows the simple columnar epithelia, simple columnar epithelia with kynocelia. And this slide is from the uterine tube. And we will have a special staining in case of this slide, which is called iron hematoxylin. So you see black color in case of this slide. It is the color of iron. And also we add hematoxylin to have a basic staining for the nuclei. We use iron in case of the slides when we want to show the smaller details, for example, kynocelia, the detail what we want to see with iron in case of this slide. I mentioned that this slide is from the uterine tube. And how can you imagine the uterine tube? The uterine tube is a very small tube, like a small canal between the ovary and between the uterus. And if we have a cross section from the uterine tube, we will see the lumen of the uterine tube. And it's very interesting because the uterine tube is has a very folded epithelium, a very folded mucous membrane. That's why the lumen will be very narrow because of this folded mucous membrane. Do you know what does it mean, mucous membrane, histologically? What is the definition of mucous membrane?
Basically, they produce the mucous materials.
Yeah, more or less it's true, yeah, but histologically, mucous membrane means the surface epithelium and the underlying connective tissue together is called mucous membrane. And in case of the uterine tube, this mucous membrane, epithelium and connective tissue together, almost covering the whole lumen, and that's why it will be a complicated view. So for the mucous membrane. So let's see this slide. Here you can see this slide. You see blue color of hematoxylin and you see black color of iron. And here you can see this folded mucous membrane with the epithelium and the underlying connective tissue. And in case of this slide, the hematoxylin has not a normal color. This color, it's a too much blue because of Photoshop, and this blue color, it's not a normal color of hematoxylin. But please don't say it on the exam because of the Photoshop. You have to say it's a iron hematoxylin staining. Hematoxylin has basophilic color, iron has black color. So let's see this slide. We have to find the surface epithelium. in case of this part, you can see very well the surface epithelium. So please describe this tissue. The first step, we have to say which type of basic tissue do we have. So it's an epithelial tissue because the cells are very close to each other, and between of them we cannot see the intercellular substance. Okay, we know it's an epithelial tissue. Let's see the function of epithelial tissue. It's a surface epithelium because it covers a surface. That's why it's a surface epithelium. Okay, next step, we have to classify the surface epithelium. We have to see the number of cell layers, and we have to see the shape of the cells. So please tell me the number of cell layers if you see this part of slide. Simple or stratified epithelia, or pseudostratified? If you see this part of slide, it's very easy to recognize in case of this part.
I guess it is stratified.
Sorry?
Stratified?
No, it's not a stratified. So
Because of too many nucleus?
It's a simple epithelia. I take a photo from this and I'm drawing the cells. This is a simple epithelia because the cells are arranged in one row, and I'm drawing the columnar cells. These are columnar cells. That's why it's a simple, and all of the cells are sitting on basement membrane, which should be here, between of the epithelial tissue and connective tissue. Here is the basement membrane, and all of these cells reach the surface. These are a simple epithelia. Simple epithelia, and the shape of the cells, I mentioned that previously. If you see the shape of nuclei, oval shaped, and these nuclei are parallel to the surface. Here is the basement membrane. Okay, and if we have a simple columnar epithelia, we always have to see the surface modification. And how can we recognize the kynocelia on the surface? How do you know it's a kinociliated epithelium, not a microvilli or not a stereocilia? So kynocelia are longer than the microvilli. That's why you can see them separately from each other, more or less. And these separate processes look like very small hairs. So you see short separate processes which are parallel to each other. Kynocelia is always parallel to each other. And what will help you, this line, it's responsible for the fixation of kynocelia. How do you call this line here? What is visible very well here under the kynocelia? This is the line of basal bodies here. In case of kynocelia, sorry, we can see the line of basal bodies. Basal bodies. Okay. And how can we recognize the cynocelia? I mentioned that they are bigger than the microvilli, and the cynocelia, because they are bigger, look like separately from each other, and we will see the line of basal bodies. And these small hairs, small processes, they are parallel to each other, more or less. Okay. And in case of this type of epithelia, there are some extra cells. Cells of this type of epithelium. The first one will be the PEG cell. And the PEG cell, how do they look like PEG cell? Sorry, here. I want to show in case of this line. Here, you see the PEG cells here, and for example here. PEG cells, as you can see, they are higher than the epithelial cells, and so darker, higher cells which don't have any cynocelia. We have several information about function of PEG cells, and these are different type of theories. But in our institute, in our department, the most accepted here, it's the PEG cells. They are degenerating secretory cells, and these were secretory cells, but they are already in the progress of the degeneration. So logical question, where are the secretory cells? We cannot see them in our slide. You should know that during the hormonal cycle, the number of PEG cells and also the size of epithelial cells, the height of epithelial cells could change. So you could see some lower columnar cells, for example here, which might look as cuboidal cells. It's just a hormonal effect. During the cycle, the cells could be higher and also could be shorter. Please don't mix it with simple cuboidal epithelium. How do you know this is not a cuboidal epithelium? Because the cuboidal cells never had cynocelia on the surface. You have several details what help you to recognize this type of epithelia. So if we check the slide, where is this slide? Here. You see the surface epithelium, simple columnar with cynocelia. And I would like to show you the PEG cells. I try to find a nice region where we can see them. Very well. I cannot find a nice region. So I show you another slide, maybe there. A moment. This is also a uterine tube with simple columnar epithelium and cynocelia. And here you see the folded mucous membrane, and you see the simple columnar epithelia with cynocelia. And in case of this slide, for example, here you see this bigger, darker cells without cynocelia. These are PEG cells. I take a photo from this. Here you can see the PEG cells. Okay, here PEG cell, other PEG cell, here is other, and here, what do you think? What is this here? Which is an extra cell. It's also lymphocyte. Lymphocyte, it's a small, it has a small nucleus, approximately it has 5 micrometer diameter, and around the lymphocyte we always have a pale zone. It's special for the lymphocyte, that's why it's very easy to recognize the lymphocyte. So here you see a lymphocyte. Okay. This is a lymphocyte. Okay. And please don't forget during the hormonal cycle the number of PEG cells and also the height of epithelial cells could change. Okay. And I have some question. Please tell me which type of cell is this that I'm showing.
This is lymphocyte.
This is a lymphocyte. Okay. And please tell me what is this? Which type of cell?
This is PEG cells because
This-
it is darker. It doesn't have kinocilia, and it is reaching the surface and longer than the side cells.
Very good. Thank you. And what is this line?
Basal body.
Line of basal bodies. What is the function of the line of basal bodies?
Body of the kinocilia is placed inside.
Fix the kinocilia on the surface of columnar cells. And what is the surface modification? How can we recognize the surface modification if you see this part of slide?
This is kinocilia since they are parallel to each other and they are we can see basal body under it, and they are bigger than microvilli.
Very good. Oh, you were your name. I can see you. Who was this?
I'm Shayan Hanife.
Okay. Perfect. I'm very happy. Okay. So, and under the superficial epithelium, the surface epithelium, you can find the connective tissue here. And in this connective tissue, we have many vessels. You see these vessels. And in the lumen of vessels, you can find the red blood cells. These are red blood cells, for example, here. Okay. And in case of this slide, there are two clinical importance that should be mentioned. The first is caused with the folded epithelium. If we see this folded epithelium, and that's why it has a very narrow lumen. I take a photo from this. Okay. This is the schematic picture. This is the real lumen of a uterine tube. And probably, you know, after inflammation, the tissues can stick together. The lumen here is very narrow. What happens in these folds? They can stick together. So the consequences can be oocyte move to the uterus. Can the sperm cell reach the oocyte? It's not possible. So infertility can be a result of the inflammation of uterine tube. Some sexually transmitted diseases could cause inflammation of the uterine tube, which can result in fertility. This was the first clinical importance in case of this slide. And the second one may be even more serious disease of consequence of this organ because of the characteristic. You can see that in the connective tissue we have many vessels. So, for example, here under the epithelium in the connective tissue we have many vessels. I take a photo from this part. You see, these are vessels. Many vessels. And so it's a highly supply organ with the blood, oxygen and nutrition. That's why the embryo could be implanted here into the wall of uterine tube, not inside the uterus. It's called extrauterine pregnancy. When the embryo is implanted here. So for sometimes it can go because highly vascularized connective tissue. What happens, and the embryo can grow and grow, and after time the uterine tube ruptures. It cannot enlarge anymore, and what happens, almost bleeding from these vessels. And what are the symptoms? In case of this disease, patient has a very bad abdominal pain. It's a very serious disease. The second one, clinical importance, was the extrauterine pregnancy, and the first one was the inflammation. Of the mucous membrane. Okay, can we move on the next slide? Okay, our next slide is from the epididymis. The next slide is from the epididymis, where we see pseudostratified, columnar epithelia with stereocilia. Okay, and the slides from the epididymis, which belongs to the male genitalia. And we have two different slides with two different staining methods. The first one is stained by hematoxylin eosin, and the second one is stained Alzan staining. Okay, and epididymis slide is more complicated because sometimes you can see other organs in the slide. So sperm cells are produced by which organ? If you see the organs of the male genitalia. The sperm cells are produced by testes. Here you see the testes. And on the surface of testes, there is a smaller organ, which is called epididymis. This organ will be the epididymis. It has several parts. The second semester, we will learn about the epididymis. It's located like this, but there should be some connection between of two organs. So sperm cells are produced in testes, inside of the testes. There are small ducts which connect the testes with the epididymis, drain the fluid from the testes. These small ducts are called efferent ducts of testis. These small ducts here between of the epididymis and testis, here, they are called efferent ducts of testis. Efferent always means something away from somewhere. Sperm cells traverse through the efferent ducts of testis and then they arrive to the epididymis. Inside the epididymis, we also have one duct which is coiled up. It's called duct of epididymis. Inside of the epididymis, we have one duct which is coiled up, and it's called duct of epididymis, and it has a Latin name. The Latin name of this duct will be ductus epididymidis. Epididymis, the organ itself, ductus epididymidis, the name of this duct. But the English name, duct of epididymis, is absolutely enough for you. This was just an interesting thing. Okay. I have to tell you this because we have different slides. One slide is taken from here. This slide is stained by hematoxylin eosin, which shows the head of epididymis. And in case of this slide, you will find two types of ducts, the efferent ducts of testis and duct of epididymis. The main thing will be able to distinguish these ducts. But there are some slides which also contain the testis part, like this. This slide will be the Azan stain slide. They will be different. Okay, and in the first semester you have to recognize the testis parts, but you don't have to know any exact details about the testis, because we will learn about the testis in the second semester. So let's see these ducts. In case of the first one, in case of the first slide, which is stained by hematoxylin eosin, we will see the efferent duct of testis and duct of epididymis. Okay, here you can see this slide. If you see this slide with small magnification, you will see two types of duct. You see a nice duct with irregular shape, regular shape, sorry, and regular epithelium, and here you see smaller ducts with irregular shape and irregular epithelium. So this is the Azan stain slide, where you see these ducts, efferent duct of testis, ductus epididymidis. So, sorry, here we should start with this duct, the nice one. If I go closer, we have to describe this tissue here. it's an epithelial tissue because the cells are very close to each other, and it's a surface epithelium because it's a duct which is lined by this type of epithelia. Okay, surface epithelium. Which type of surface epithelium? We have to see the number of cell layers and the shape of cells. Here, the nuclei are in several layers or are in several levels, but all of the nuclei will be oval-shaped, perpendicular to the surface. That's why it's this epithelium. It's a pseudostratified columnar epithelia. And in the basal part of epithelia, you see these around this cell. They will be the reserve cell, what we mentioned previously. And if we have a pseudostratified columnar epithelia, we have to see the surface modification. Here you see the surface modification. It's a stereocilia. How do we know it's a stereocilia? Because a huge surface modification, what we see on the surface, they are very long, and if you see them, they are not parallelly to each other, they are attached to each other. That's why we know these are stereocilia on the surface. It's a pseudostratified columnar epithelia with stereocilia. Okay, and these cross-section of duct will be the cross-section of duct of epididymis, and these will be the nice with regular shape and with regular epithelium. And these are the cross-section of duct of epididymis. Okay, and let's see the smaller ducts, the ugly ducts. They will be irregular, with irregular shape and irregular epithelium. The epithelium is can be simple columnar and pseudostratified columnar epithelium in same time, and on the surface of columnar cell we can have stereocilia or we don't have stereocilia. It's very variable. That's why we will see this very irregular epithelium, it's very irregular shape. And these ducts are the efferent ducts of testis, which are located between of the testis and epididymis. And around these ducts, you see connective tissue with the eosinophilic color, light eosinophilic color here. And if I move closer, you see some different cells with more darker eosinophilic color here. These cells will be smooth muscle cells around the ducts. For example, here, or here is also visible, these are smooth muscle cells and they have darker eosinophilic color than the connective tissue. Okay, I'm writing these details here. Stereocilia, they are attached like this, and stereocilia is a huge surface modification structure, and we can see the reserve cells. That's why it's very easy to recognize the pseudostratified columnar epithelia. And around the vessels here, this color, these are smooth muscle cells with darker eosinophilic color. And here you see the color of loose connective tissue. Okay. And in this zone, these are the efferent ducts of testis. Shows the if ducts of testes, and for example, in this zone, you can see the cross-section of duct of epididymis. Okay, and let's see the epithelium again. Between of the columnar cells, sometimes we can find lymphocytes. Okay, like an extra cells, lymphocytes. Okay, let's see this slide. Okay, please tell me which type of duct is this? The nice or an ugly?
Abnormal, ugly.
This is an ugly.
Not really.
Not really. It's a nice one, because if we see the surface epithelium, which type of epithelium do we have here?
Pseudostratified.
How do you know it's a pseudostratified columnar epithelia?
Because of the reserve cell.
The first point, we see the reserve cells, which are special for this type of epithelia, and the shape of nuclei, all of them have oval-shaped columnar, oval-shaped nuclei, and these nuclei perpendicular to the surface. And what is the surface modification here? Stereocilia. How do you know it's a stereocilia? Not a microvilli, not a kinocilia. How can we recognize the stereocilia on the surface? Stereocilia is the biggest surface modification. It's a huge structure, and they are they don't stand parallel to each other. If you see this picture, you can see them stereocilia are attached to each other. Okay. Which type of cells are here in this zone with darker eosinophilic color?
They are smooth muscles.
Smooth muscle cells. Okay. And which type of tissue here?
Loose connective tissue.
Loose connective tissue. Okay. And which type of duct here? What is this? The nice or ugly one?
I think this one should be ugly one.
And if I'm going...
Sorry, how can we distinguish the ugly one and the nice one? I missed that part.
The nice one, I mean, the nice one has regular shape, more or less roundish, and the surface epithelium, it's very regular. It's a normal pseudostratified columnar. And if you see the ugly one, the ugly will be more or less, most of times, they will be smaller than the nice. And if I'm going closer, you can see the epithelium. This epithelium, it's very variable, can be simple, can be pseudostratified in the same time. And on the surface of columnar cells, we have stereocilia or we don't have stereocilia. That's why it will be very irregular. The epithelium is very irregular. This is the difference between of the nice and the ugly. In case of this epithelium, we cannot see very well the reserve cells and the pseudostratified columnar epithelium. We cannot see the stereocilia on the surface very well. That's why it's an ugly one. And what do you think this structure? It's a small repetition. What about this? What is
I
this?
think it is a vessel.
Very good. It's a vessel, and in the lumen of vessel, you can see the red blood cells. Okay. And I have a tricky question. So, one moment. What about this? What do you think? Sometimes you see this. What is this? Sorry, I make a photo from this.
Isn't it the degrading cells?
No, it's just a tangential cut. Tangential cut. If we make a section, for example, from here, we will see this picture. We only see the columnar cells. It's called tangential cut. It's a tricky question. We have a this is written to the level of cut and the direction of cutting. We can see this picture of the duct. Okay, and I show you the second slide from the epididymis. One moment. The theoretical material will be the same, but we have to see the staining methods. The staining methods will be different. Azan staining, carmine aniline blue, connective tissue fibers will be blue because of the aniline blue. And the other parts of the slide will be red. And if we see the nuclei, all of the nuclei have red color. That's why we have to be sure this is an Azan staining. Okay, let's see the ducts here. What about this? This is a nice one or this is an ugly one? If you check the surface epithelium.
This is a nice one since all of them are in the same size.
Yes, we can draw the lumen with one line, one circle. That's why it has a regular epithelium. And here you see the reserve cells with roundish nuclei. This is a duct of epididymis. Okay. What about this part?
It's a loose connective tissue.
It's a loose connective tissue. And what is this here?
That's vessel.
Okay. And what about this part?
That's tangential cut.
Very good. And for example here?
That's ugly one.
And the name of the ugly?
Duct of epididymis.
The duct of epididymis is the nice one. Efferent.
Is an efferent duct of testis.
Very good. Okay, efferent duct of testis. Okay, these ducts are the same what we see previously. And in case of the other part of slide, you can see a small part of testis. If I'm going closer, you can see the tubules of testis, and these tubules are lined by a special stratified epithelia. And in this special stratified epithelia, you can see the immature sperm cells. And in the first semester, you have to recognize this is a testis part, but you don't have to say any details about the testis because we will learn the testis in the second semester. Okay, so practice. Let's see. The surface modification. What is the surface modification here?
Stereocilia.
Stereocilia. How do you call this cell?
That's a reserve cell.
And the function of reserve cell?
They are for regeneration.
Very good. Which type of cell are around the ducts? It's not so nice visible with ozone staining, but these cells should be
Smooth muscles.
Smooth muscle cells. Here, which type of duct is this?
That's the efferent duct of testis, obliquum.
Very good. Okay, and my last question is, so what about this cell between of the columnar cells with roundish nuclei? I mentioned this cell previously.
They are the degrading cells.
No, it's a lymphocyte.
Oh, lymphocyte, yes.
It's a lymphocyte with roundish nuclei, and around the nuclei, more or less, we can see this paler zone. It's a lymphocyte. We can find lymphocytes in case of this type of epithelia as well. Okay. Questions about the pseudostratified columnar epithelia with stereocilia? Okay, and we have another slide. The next slide is pseudostratified columnar with kinocilia. I make a photo from this. And our note will be complete. An ozone stain slide where you can distinguish, you can compare the nice one and the ugly one. And in case of this slide, you can see a small part of testis, which is visible here. And testis tubules, they have special stratified epithelia, which contains the immature. Okay. And here it's a testis. Okay.
Excuse me, what was that dots that you mentioned? The
Sorry?
red dots.
Sorry, I can hear you.
This you mentioned these red dots are the place that the sperm immature sperms are located. What is the name?
There are several immature phases, and they have several names. Spermatogonia, primary spermatocyte, secondary spermatocyte, but in this semester you don't have to know these names. Immature.
So just you need to say immature.
Yes, absolutely enough. This part is the testis, and here you see the duct of epididymis, the nice one. This one, for example, this one and tangential cut of it. And here you see the ugly one, the efferent ducts of testis. The color was green, and here you see the ugly ones, for example, here. Okay, and now our note is ready. Okay, the next slide, which shows the pseudostratified columnar with cynocelia. This slide is from the epiglottis, and this slide is stained by PES age. Okay, and epiglottis belongs to the respiratory tract, and that's why in the respiratory tract we will see the respiratory epithelium pseudostratified columnar with cynocelia. So as you will see several types of tissue in case of this slide. I'm showing you one moment, just a minute. Here you see this slide with several tissue. So the first task is to find the side where we have epithelium. This is the side where we have epithelium. And first we will describe the epithelium. And previous that, what is the staining? I mentioned that it's a PES age staining. What is the dominating color? What is the extra color? This purple color is caused by PES staining. The staining is PES age. PES staining combined with hematoxylin. We use hematoxylin to stain the nuclei. If I'm going closer, you can see the nuclei with basophilic color. The first task, we have to describe the epithelium. Sometimes you will see that the slides are not very nice because this is, you know, after the death, degeneration process is starting. That's why sometimes the surface epithelium is not so beautiful, but more or less you can see the type of the epithelium here. These cells are very close to each other. It's a surface epithelium, and you see here the reserve cells. And you know, the reserve cells, they are special for the pseudostratified columnar epithelia, and they are responsible for the regeneration. And if we have a pseudostratified epithelia, we have to decide what is the surface modification. What we see, please tell me, what is the surface modification theoretically? I mentioned that it's a cynociliated, but and how can we recognize the cynocelia? I know I asked this question several times, but
because they are parallel, and we have basal bodies on them.
We see them separately from each other, that's why bigger than the microvilli, and we can see the line of basal bodies. For example, here you see the line of basal bodies very well. That's why we can recognize the cynocelia. The function of cynocelia?
Or for cleaning the air.
They can move, synchronized, yes. All dirt will stop in the mucus, and the cynocelia will move the mucus upwards. It has a key function in the respiratory tract. they move towards your nasal cavity and towards the oral cavity. So let's see the extra cells of the epithelium. I make a photo from this part. This is the epithelium. Here you see the cynocelia. And you see the line of basal bodies here. Oops, here. Line of basal bodies. We see the cells, extra cells, and then we have to stop because it's time. Okay. The extra cells. Here you see these PES-positive cells, PES-positive cytoplasm. It's a very thin cells. They are goblet cells. Again, different like previous goblet cells, but they are also goblet cells. Goblet cells. You see here the goblet cells with PES-positive cytoplasm, because the goblet cells secrete mucus. That's why they will be PES-positive. There is a very special to respiratory tract inside the epithelia. Inside the respiratory tract, we have a very thick basement membrane. You see this thick basement membrane here. This is a basement membrane, and this basement membrane is very thick in case of the respiratory tract, and the basement membrane is also a PES-positive because of the reticular fibers. The basement membrane will be PES-positive. It's very important. And what about these small round cells? For example, here. Which type of cells is this?
Is it reserve cell again?
No, the reserve cells should be the basal part of the epithelia, but these cells are between of the columnar cells.
And lymphocyte?
Lymphocytes.
Sorry, I missed a part. What is goblet cell?
Goblet cell?
Yeah, goblets.
Goblet cells belong to the glandular epithelium. We will learn about later. And these goblet cells are embedded between of the columnar cells. And the cytoplasm of goblet cells will be PES-positive because the goblet cell produce mucus, and the mucus contains many carbohydrates. That's why the goblet cells will be PES-positive. They belong to the glandular epithelium. Here you see this purple cytoplasm. This purple cytoplasm belongs to the goblet cell. For example,
are they as long as the columnar cell?
They have the same size. But I know it's a complicated view because there are many cells which are very close to each other, and that's why you cannot see the real size of these cells. And the last extra cell that you should recognize here in one row around this cell. These are the reserve cells. Okay. I think we mentioned all of the extra cells what you have to recognize in this epithelium. Do you have any questions, more questions about this slide? We will continue with this slide next time. Questions?
No, thank you.
Okay. I should stop